Good day, Dr. Shu, Dr. Khalidah, Dr. Azma, and my fellow classmates. So today I'm going to give my presentation titled Extirpation of Orconectes propinquus via the hybridization with Orconectes rusticus. The freshwater ecosystem is naturally very prone to hybridization and integration, and this is also the case for the crayfish populations. What makes it important to study is that crayfish species are considered as keystone species in the freshwater ecosystem, so we should understand why the extirpation of Orconectes propinquus is happening via the introduction of Orconectes rusticus and the hybridization that's occurring. The Orconectes rusticus is actually endemic to Ohio River and its tributaries. But then by year, it got spread. Whenever this Orconectes rusticus got established, it has a track record of extirpating the native crayfish species. So several states actually put restrictions and bans to the usage of Orconectes rusticus as fishing baits. Methods and materials. So, the paper by William Perry et al. In the year 2001 had actually two kinds of trapping procedures. The first was to actually do a population survey of the crayfish species in the trout lake to actually study how far the spread of Orconectes rusticus is there. So this was done using the standard trapping procedure, and it was done annually in August from the year 1990 to 1998. So what they did was that they actually trapped the crayfishes and then identified their sex, measured their carapace length, identified the species, and then released them out. The second trapping procedure was to actually study and assess the movement pattern of the Orconectes rusticus. And what they did was mark, release, recapture experiment. Genetic analysis. The first thing they did was to study the IDH and HAD allozymes. They used the standard horizontal starch gel electrophoresis to do this. They used the samples from the population survey trapping procedure in the year 1995, and they wanted to actually see the existence and the distribution of hybrids in the trout lake. The second procedure of genetic analysis is actually a restriction fragment length polymorphism analysis of mitochondrial DNAs of the crayfish species. So what they did was that they went to the Orconectes rusticus home range allopathic site collecting the species, to the Orconectes propinquus home range allopathic site collecting the species, and studied for the specific restriction site, especially the VSPL and DAQL restriction sites in their genes. So the third genetic analysis was actually to know the mode of inheritance for the mitochondrial DNA. What they did was that in May 1997, they collected buried females, means they have eggs, and were transported back to lab using separate plastic containers and were put into water flow system. So once the eggs were hatched and the juveniles reached stage 2, the mother and 10 of their juveniles were scored for allozymes and mitochondrial DNA restriction sites to know the mode of inheritance. So next is the estimation of crayfish fitness, and they had two. The first is the study of the female fecundity and the juvenile survivorship. They took these 116 mother and juvenile families from before. We just counted the composite index to study the female fecundity and the juvenile survivorship. At last, to know the competitive abilities of the crayfishes. So what they did was that 24 hours of starved crayfish was used, and a pair of crayfish will be placed in the pens for the seeing of the relative competitive abilities. And the trial started 1 hour after the lab's lights were off, and a 1.5 cm of nightcrawler was placed in the middle of the pen. And this experimental arena was inspected every 10 minutes with a red light until one of the crayfish possessed the nightcrawler. So after everything finished, the crayfishes were actually identified morphologically and genotyped as well. So the next paper is by Arcola et al. In the year 2014. So they only studied the growth, survival and the fertility of these crayfishes. The methods and materials are almost similar to the collection of the buried females from the previous paper, with some distinctions, whereby in this paper they collected the buried females from overlap zones in the trout lake, so that there's a maximized number of females carrying a hybridized offspring. So juveniles hatched, and 25 of these juveniles were taken from each mother and frozen, as well as the mother after calculating the abdomen for actually doing the composite index like before to know the fertility of the crayfishes. And then the mother was frozen as well, and they were frozen because to study the allozyme like before and to identify the species. But the rest of the juveniles were left to survive to actually calculate the growth and the survival shapes of these crayfishes. And now results. So the spread of the Orconectes rusticus in the year 1990, almost virtually everything Orconectes rusticus. As for the movement pattern is that they like to go slightly more to the south rather than the north, and almost everything in Alecrush Creek itself, because in the south is actually more rockier and attractive to the crayfish. There's actually a diffused hybrid zone approximately 2 to 10 km from the Alecrush Creek, and very important to note is that most of it are backcrossers. As for species-specific mitochondrial DNA haplotypes, from the allopathic side, the restriction site of VSPL and DAQL in Orconectes propinquus is only at 16S rDNA locus, which could not be found in Orconectes rusticus. And for Orconectes rusticus, these restriction sites can only be found in the cytochrome C oxidase 2 genes, which could not be found in Orconectes propinquus. So when they studied the mode of inheritance of mitochondrial DNA in the hybrids, they actually found a maternal inheritance. And that's actually a preference of Orconectes rusticus females to mate with Orconectes propinquus males. So what happens is that a cytonuclear disequilibrium. 93.6% of the hybrids actually has the Orconectes rusticus mitochondrial DNA haplotypes. This is due to a strong mating asymmetry, but we do not know why this is happening. The first paper actually studied the crayfish fitness and found out that the fecundity and the juvenile survivorship weren't significant among the species. Going to the next paper, the fertility weren't significant among the species, but the latest survivorship of the juveniles were very significant, and most in the F1 hybrids, and the least, note this, the least in the backcrossers. Okay, moving back to the first paper, they actually studied the competitive abilities among the species and found out that the F1 hybrids are superior, and in the second paper, the growth of the F1 hybrids were the highest. Discussion. We need to acknowledge two paradoxes. First is the females, Orconectes rusticus, mates with the Orconectes propinquus males, but this should slow down the hybridization and extirpation because the females mates with one male and the males mate with many females. The next paradox is the F1 hybrid viger. If there is a hybrid, how does the Orconectes propinquus extirpates fully? Why there's no hybrid zone? First is the F1 hybrid viger, because Orconectes rusticus comes in and then extirpates the Orconectes propinquus. So with the hybrid vigor that the F1 hybrids has, it combines strength with the Orconectes rusticus and attacks the Orconectes propinquus more. The next point is the backcross hybrids. So basically the F1 hybrids, they tend to backcross with the Orconectes rusticus, and the later generations tend to reduce their fitness level, causing to don't have any hybrid zone, and the Orconectes propinquus, despite being extirpated as a genotypically distinct entity, due to these backcrosses, the traces of these Orconectes propinquus, the genetic traces in the lake, is also fully extirpated from the lake. And third is the continuous introduction and invasiveness of Orconectes rusticus, because when the hybrid later generations are very weak, this Orconectes rusticus that keeps coming in keeps invading and pushing the invasion front forward. So the net outcome is the extirpation of Orconectes propinquus. Concluding note, I would like to say that even though these two crayfish species prefer a similar niche, they are not ecologically equivalent. So from a conservationist point of view, this issue should be looked into and not taken slightly. Thank you.